I'm delighted to introduce Dr. Leena Malayil, who will be moderating the session.
So I'm Dr. Leena Malayil. I'm an assistant research professor in the Department of Global Environmental and Occupational Health. And I'm one of the co-PIs of the Global Future Alliance and the co-leads for the Nepal Future, along with Sri Krishna Dattal. I also serve as the co-director for the Maryland Safe Drinking Water Study, along with Dr. Leena Malayil, assessing the microbial quality of drinking water in underserved Maryland communities. I'm extremely delighted to moderate the session, the trainee and the early career scientist lightning round. We have 9 speakers among us. So I'm going to call up the first speaker, who's also my close colleague, Dr. Michele Morgado. She's a postdoctoral associate with the Department of Global Environmental and Occupational Health at UMD School of Public Health, where she received her PhD in environmental health science. Her research focuses on understanding how to better protect public health from the increased risk of bacterial, waterborne, and foodborne infections with climate change and increased environmental stresses, as well as global health-related issues within the food, energy, and water nexus, with a focus on interdisciplinary and transdisciplinary research and collaboration. So welcome, Michele.
Good afternoon, and thank you, everyone. So today I will be changing it up a little bit, talking about a foodborne, an important foodborne waterborne pathogen, so Vibrio, and I will be presenting a summary on a long-term analysis of 2 Vibrio specifically, so Vibrio vulnificus and Vibrio parahaemolyticus, and how their virulence factors may be associated with environmental changes over time in the Chesapeake Bay, Maryland, in the US. And this was a collaboration between the Environmental Microbiology Lab, run by Dr. Amy Subkota, and Dr. Rita Caldwell's Microbiology Lab. So the main objective of this study was to evaluate long-term trends in Vibrio virulence factors associated with pathogenicity in the Chesapeake Bay, Maryland waters across 2 3-year sampling periods. So 2009 to 2012 and 2019 to 2022. So this topic is really of public health significance because we know that the incidence rate of Vibrio infections, known as vibriosis, has been increasing over time in the United States. For example, between 1996 and 2018, the incidence of Vibrio infections increased from 0.15 to 0.68 per 100,000 people. And according to CDC data, unlike other bacterial pathogens, for example, Campylobacter, Listeria, Salmonella, Vibrio infections have steadily increased over time, especially in the last decade or so. We know that there are roughly 80,000 cases of vibriosis reported in the United States each year, but this may be an underrepresentation, especially for those less severe cases. One of the leading causes of shellfish-borne bacterial illness in the United States is Vibrio parahaemolyticus, and its overall abundance is known to be strongly associated with water temperature. Another pathogen, Vibrio vulnificus, is the leading cause of mortality via water and seafood exposure. And the environmental trends for this Vibrio bacteria are less understood. The important thing here is that for these 2 Vibrio and other Vibrio bacteria, there's a really strong correlation with environmental changes, including things like sea surface warming and extreme heat events. And so it's important to look at these associations and trends in important large estuarine systems like the Chesapeake Bay. So for this project, we applied what's known as a retrospective longitudinal molecular analysis, and we looked at 2,415 isolates of Vibrio parahaemolyticus and Vibrio vulnificus, which were recovered from 14 stations across the Chesapeake Bay in the Maryland side of it. So we didn't go all the way into the Virginia side. And this was done, so these were isolates collected as early as 2009 and as late as 2022. We performed DNA extractions, PCR analysis of virulence genes and performed ANOVA, pair t-test, CRISPR-Wallace regression analysis. Some of our interesting findings include the distribution of where most of the isolates with virulence genes were located. So just to give you an idea, the TDH and the TRH below, these are the virulence genes for Vibrio parahaemolyticus. And we found that most of these isolates occurred in the upper parts of the Chesapeake Bay, so closer to the Susquehanna River. For Vibrio vulnificus, the virulence genes that we looked at were VCGC, PIL-A, and RTXA. And we found that most of these isolates were more distributed in mid areas of the Chesapeake Bay and lower areas, so as low as Tangier Sound, so closer to the mouth of the bay. We also really wanted to see if there was a strong association with environmental parameters like temperature and salinity. There ended up not being statistical significance, but importantly, we did find that there was a trend. For the parahaemolyticus, the temperature threshold was closer to 25 degrees Celsius, where the cluster of isolates was located, and salinities were lower. And then for vulnificus, the temperatures were higher, as high as 30 degrees Celsius. We found most of the isolates, and salinities were higher as well, so as high as 25 parts per thousand. Major conclusions, we found that potentially pathogenic Vibrio occur across the bay throughout the year, including in times where we did not think we would find as much, so fall and winter. Vibrio vulnificus isolates had greater changes to virulence genes over time compared to parahaemolyticus, and there was a greater prevalence of Vibrio vulnificus isolates with virulence genes in mid to lower sections of the bay. This is important because it tells us that we need to improve awareness of risk of severe vibriosis from wound-associated water exposure, especially through targeted educational outreach among recreational and commercial users, especially in those mid to lower sections of the bay where you could be more susceptible to that. And it's also important to note prompt treatment in more vulnerable populations, which includes males over 45 with underlying medical conditions. And just some acknowledgements of our funding and colleagues and mentors throughout the project. Thank you, everyone.
Thank you, Michele. Next, we have Dr. Nathan Boyd along with Jerin Tasnim. Nathan Boyd is a postdoctoral researcher at the UMD Clark School of Engineering, leading research on flood, disaster aid, infrastructure, and social impacts. He also collaboratively works with multidisciplinary team investigating the social and environmental impacts of septic tank flooding near the Chesapeake Bay. So joining Dr. Boyd again is Jerin Tasnim, a graduate student in the same department.
Everyone, today we'll be talking about understanding the impact of sea level rise to septic systems and other wastewater alternative management strategies in the Chesapeake Bay region. Okay, so the overview of this work is that, of course, as we know, climate-induced flooding and sea level rise is going to impact the Chesapeake Bay. And for septic systems, there needs to be a gap between the groundwater and the drain field so that the waste can percolate through unsaturated soil, which is where most of the waste is converted to a more benign form. When this doesn't occur, there are environmental consequences, so discharges of nutrients to the environment. And also there could be public health consequences for flooded drain fields, and bacteria can impact communities. So another thing that we know and find is that in Maryland and on the Eastern Shore specifically, septic systems are very prevalent in rural communities that are also socially marginalized as well. So there's the increased likelihood of problems, but also increased consequence. And so that motivates our work. So our research agenda to tackle this problem is to first, we want to quantify the problem. So our first research question is, how do wet weather events, so that could include rainfall, riverine flooding, and then coastal flooding from sea level rise, how can that influence septic maintenance and pump out calls? So of course, when there's an issue with the septic tank, you're more likely to get calls to try to correct the problem. So that's the first part of the research agenda. And the second is, given what we know about the problem, what type of solutions, what can these affected communities do to better manage wastewater while facing flooding-induced septic failure. So for these research questions for now, we have two different study areas, and a lot of that has to do with data availability for now. So for research question 1, we're focusing on coastal Virginia because of the availability of statewide septic maintenance data that we don't have yet in Maryland. And then for research question 2, we are focusing on the Maryland Eastern Shore because there's a lot of information available on who's on septic and who's on sewer, and then what can be done for those communities who are on one or the other. So once again, our study region for research question 1 here, this shows coastal Maryland. You can see here that the red and orange points on the map are septic feet or septic tanks within the tidal zones. You can see there's quite a lot in coastal Virginia and at the end of the Delmarva Peninsula, in particular. The other light green dots are just other septic tanks throughout the coastal region. And what we know is that the coastal area of Virginia is only 29% of the overall state, but it has 60% of the state's population. Okay, so for research question 1, what we did is took time series of the flooding and the septic maintenance calls and did some transforms to identify the frequency of occurrence. And then what we did is we did analysis to show that the seasonal trends, when you take these out, that there is a strong association between the number of septic calls and the flood events, but you have to do to really separate out the different frequency bands to account for seasonality. So for the second research question, we have a number of different alternatives. The biggest thing is trying to decide between upgrading a septic tank and then putting in some kind of small diameter sewer system. And in the Eastern Shore of Maryland, you can see here that the different, each dot represents a different type of septic tank. So one that's within a sewer service area, one that's not, is shown in either blue, but that could be where septic or sewer can be extended. And then the gradient of orange to red are places that are would have to build a completely new wastewater management system on their own. So there's different cost implications for different parts of the state. And so the main takeaway is that each community needs their own strategy. And so there's going forward in the future, probably a large statewide and regional effort to design new wastewater alternatives to cope with these changes. Yep. And then the conclusions essentially reiterate what we said here, and I'll open it up to any questions.
So next we had Kevin Tarwa, who is a second-year PhD student in the food science program within the UMD College of Agriculture and Natural Resources. His research focuses on the sanitation of pre- and post-harvest water that is going to be used for application in growing and washing fresh produce. So let's welcome Kevin Tarwa.
Good afternoon, everyone, and thank you all for attending today. So today for my presentation, I'll be talking about the sanitization of harvested rainwater, sort of following up with what Emmy had discussed earlier in her presentation. And this project is in collaboration with, it's a multidisciplinary approach to this project, and it's led by Dr. Rachel Rosenberg-Goldstein. So just to give you guys a brief background, so harvesting rainwater can definitely be a valuable resource for farmers in terms of irrigation, especially with those farmers in areas with limited access to water source. However, harvested rainwater is a potential reservoir for human foodborne pathogens and must be treated prior to application of fresh produce irrigation. And as Emmy had showed you guys, these IBC totes that were collecting the harvested rainwater they were under a sort of gutter system. So in that gutter system, there can be a possibility where we have bird fecal contamination that could sort of trickle into that rainwater. So treatment is definitely necessary prior to irrigation. So here we evaluated peroxyacetic acid for sanitization of harvested rainwater. at 2 farms in Maryland over multiple growing seasons over 2 years. So with this being said, this project is still ongoing and we're still currently working. So for the experimental design, it's sort of a 2-part design where we first did a validation study to ensure that we have the proper concentration of PAA to essentially produce a 3 to 4 log reduction in contaminated water. So what we had first did, we collected the rainwater on site. So we did this, we did 2 trials at our first peri-urban farm in Upper Marlboro, Maryland. And then we did 1 trial so far up in our urban farm at Plantation Park Heights in Baltimore. So the rainwater was collected and we equilibrated the water to 12 and 32 degrees Celsius just to sort of capture that fluctuation that might occur during growing seasons. And then nextly, we inoculated the rainwater with around 7 log CFU per mil of a 7-strain cocktail of either E. coli or Salmonella. And these strains were provided from the EPA water testing panel that's specifically designed for agricultural water testing, in which we treated the rainwater with 8 ppm of PAA for about 5 minutes. And then we plated it onto tryptic soya agar supplemented rifampicin because the strains were rifampicin resistant and incubated for 22 to 24 hours. And this was done, just to mention that this was done in the lab scale where it was more of a controlled environment, as opposed to our verification study design where we actually did it on the farm sites, where we treated the rainwater barrels with 8 ppm of PA for 5 minutes. And then we confirmed the PA concentrations using titration kits, followed by plating the samples onto TSA. And this was repeated on day 7 as well, because we really wanted to assess how stable that PAA was in the treated water system. And just to quickly go through the results. So on the left side is our validation study. So at the top is the levels of Salmonella after following 5 minutes of PA treatment. And on the bottom is the E. coli cocktail mixture. And we're seeing similar trends at both temperatures where we are seeing about over a 4 log reduction with the PA treatment. However, on the right side with the verification study, we noticed a different trend here. So we noticed that at the top, directly treating with PAA. For the first trial, so UM is Upper Marlboro, and then PPHUF is the Plantation Park Heights farm. We noticed that we weren't seeing really a significant reduction right away on day 0. However, when assessing the PA stability over the course of 7 days, we're not really seeing any significant growth in the treated water. So overall, with the results, we noticed that in the validation study, we are seeing a significant reduction for the PA treatment. However, for the harvested rainwater on site at the farm, we noticed that there was no significant decrease in the natural microflora in the water. So one of our next steps is sort of doing microbiome analysis of the water, looking at, because as of right now, we just did aerobic play counting, so we don't really know what's necessarily in the water right now. So doing some microbiome analysis, we can get a good sense of what was present in the untreated rainwater, and then also whether there was any microbiome shifts in the water following that PAA treatment. And just for my acknowledgements for this project, this was supported by the Agriculture and Food Research Initiative competitive grant. Special thanks to Dr. Ron Tikar, my PI, and also some of the members that I collaborated with this project, with the Rosenberg Goldstein Lab, Dr. Shirley McAuliffe's lab, Dr. Manan Sharma and Dr. Alan Gutierrez at the USDA, and then of course Tom Murphy, Guy Kilpatrick, and Farmer Chippy for their help on this project. So thank you.
Next, we have Rafian Aziz. He's a master's student in the Bioenergy and Biotechnology Laboratory within the Department of Environmental Science and Technology at the University of Maryland. His research interests include sustainable waste management through food waste composting, and anaerobic digestion, with an emphasis on reducing greenhouse gas emissions. Rafian is also dedicated to exploring how composting can contribute to soil health, and water conservation, particularly in climate vulnerable regions, while developing solutions that foster environmental resilience and support community well-being. Welcome, Rafian.
Thanks, Leena, for the introduction. Today, we're going to talk about animal waste management technologies in Maryland, their distribution benefits, and their farmer perspective. The current animal technologies that we have only includes anaerobic digestion, pyrolysis, composting, and manure injection. So each of the different anaerobic animal technologies have different substrates. We use different substrates because of their time, energy, and efficiency. So let's look at these animal technologies one by one. First, we have anaerobic digestion. And Dr. Lansing and Morin talk about it already. It's basically the same process. We take the animal waste, put it in the digester, and where inside microbes break it down to biogas, a mixture of methane and carbon dioxide. And after the biogas is produced, we put them in different treatments to remove harmful gas such as hydrogen sulfide. From there, we put it in the energy converter and produce renewable natural gas. But after the digestion is complete, we have this solid and liquid nutrient-rich material that we can apply directly to the lands. But all the animal waste are not the same. If you consider poultry litter is usually dry and has less moisture than cattle manure and dairy manure. So we usually go for the thermochemical processes. I mark pyrolysis and gasification here because it requires higher temperature and low amount of oxygen. And unlike the other thermochemical processes, it produces heat, syngas, biochar that we can reuse and reapply to our agriculture. And everyone knows about composting. The key difference between composting and other technologies that it requires aerobic process, meaning it requires oxygen to complete their whole cycle. And unlike the other two processes, while it can produce biogas or energy, it produces stabilizing nutrients and kills pathogen and gives us a soil amendment that we can apply directly as a fertilizer. And last but not least, we have manure injection. Instead, applying the liquid manure directly at the soil surface, we usually inject them at the subsurface level. The reason why we do this, because if we apply directly the liquid fertilizer or manure in top of the soil, it can offset the runoff and create ammonia volatilization and pollute the nearby water bodies in here. And we don't want that. So if you look at the map of Maryland, in this map shows different concentrated animal feeding operation sites marked by the round circles, and the X indicates the different cluster zone of this feeding operation site. So larger the X, the greater the concentration of these feeding operation sites. And if you look at the density of this feeding operation site compared to the different animal waste technologies indicated by the diamond circles, you can see that the proximity and the density is really low compared to the feeding operation site. So to find the gap, we asked our farmers a simple question. What was their thought about the animal waste technologies in next 5 years? So around 6 to 10% did not support the animal waste technologies, while most of them showed strong or moderate support to these technologies. But around 26 to 32% of these farmers said they are unlikely to adopt but strongly support this kind of technologies. What this indicates that there is room for outreach, education, and provide incentives to these farmers to adopt these technologies. For the next steps, we are planning on outreach and education, and we have already published fact sheets in our website and extension, and we are publishing our fact sheet in different languages, and hopefully we can remove the barriers between farmers, policymakers, and research in the future. Thank you.
So moving away from UMD speakers, now we have Dr. Sumanta Chatterjee, who is currently working as a faculty research assistant at the Department of Environmental Science and Technology. He has a PhD in soil science from the University of Wisconsin-Madison, USA. His research areas include soil health, water-carbon coupling, precision agriculture, and agroecosystem modeling, AI and ML, and remote sensing. So I would like to welcome Dr. Sumanta Chatterjee. Thank you.
Good afternoon, everyone. So today I'll be talking on integrating satellite imagery with weather variables for enhanced cover crop biomass estimation. So recently, like most of the work, they're focused on mostly like spectral indexes from satellites, but we tried to improve the biomass estimation model by including other data like weather and other parameters. I'll talk more as we go on. So to know a little bit about cover crops, during the last decade, the cover crop enrollment in Maryland has increased double, and Maryland has one of the most like highest percentage of cover crops in terms of like area. And cover crops consist of many species, including like cereals, legumes, grasses, and mixes. So it's grown between the crops and it's kept in the field to it's not harvested to increase the soil organic carbon and then returns like carbon and nitrogen to the soil for the subsequent crops. So, and cover crops produces it provides a lot of ecosystem services, like in terms of water quality and then soil health. It reduces erosion and then increase water infiltration, improve the soil health by increasing the soil organic matter and soil organic carbon in total. So it also provides like nitrogen and carbon to the subsequent crops. So it has like carbon credits and nitrogen credits, which is very important to reduce the for stakeholders the budget of carbon and nitrogen. Cover crop performance depends on different factors, but mostly it's like soil, climate management, and the cultivars, genetics. And so on this background, we developed a methodology. The work has been done on USDA Beltsville Agricultural Research Experimental Station, which is in Beltsville. And then we collected cover crop samples for like 4 different cereal monoculture, critical wheat, rye, and barley in 4 years during like fall, winter, and spring seasons. And we used harmonized sentinel Landsat data, HLS data for NDVI, which is the main predictor here for biomass estimations. NDVI is actually a ratio that gives you like whether the vegetation is healthy or not. So it ranges from -1 to 1. 1 meaning the like higher like healthy vegetation, and negative meaning there's like barren land or no vegetation. But NDVI has some issues, like it has saturation problem whenever the biomass is like more than 1900 or 2000 kg per hectares. The satellite signal get like saturated, and then there is difficulty to estimate beyond 2000 limit. So we tried to improve upon these issues to include like other variables, whether that could improve the model further to estimate like higher ranges of biomass. So we included growing degrees, which is a popular weather index, which is actually a thermal index. It accumulates the degree days, and it's related to the phenology. Like we have more degree days, we have like faster phenology. And we also included photosynthetically active radiations, which is also another parameter for yield estimation. So to talk about the model that we in this graph, we see that the different season and different year, the cover crop performance varies. Like if we talk about dry biomass, we can see that during the different season, it is like different, it varies differently. Same like goes for NDVI. And we then included different models here. One is like only remote sensing based, and then one is weather based, and then hybrid based. We found that compared to the remote sensing based, if we only use NDVI, the hybrid model performance like improved better with R-square of 0.80 and RMSE of 600, which is quite good compared to the only spectral index. And then we used also machine learning models, and we found that the decision tree based algorithm like random forest perform better than the boosting algorithm like XGBoost, stochastic gradient boosting, etc. And then we also did a temporal generalizability of the hybrid model to see how the model performs in different years. And we saw that they're performing really well for different years. So it has temporal generalizability. So to summarize, I can say that we can use weather-based we can integrate NDVI and weather host model to increase the performance of the biomass estimation models. But we also can use the machine learning models for to scale up the data. So this model has some future implications. So it can be used in cover crop nitrogen calculator to calculate the nitrogen and carbon credit for the subsequent crops. So this data can be used, or the model can be used for initial app development for cover crops. So with this, I want to thank my funders and my collaborators. Thank you.
Dr. Chatterjee. So next we have Dr. Gibson Cisco. He's an assistant research professor at the Maryland Energy Innovation Institute at the James Clark School of Engineering. His research focuses on ionic liquid-based catalytes for quasi-solid state batteries, interfacial impedance reduction, and electrochemical impedance spectroscopy modeling.
Hi, everyone. Thanks for having me here. So yeah, I'm Dr. Gibson Cisco, and today I'm just going to be talking about this battery prototyping lab at the University of Maryland. So this is a pretty new facility. It got brought up about a year ago, and it's a collaboration between the Material Science and Engineering Department and the Maryland Energy Innovation Institute. So the goal was, when the Inflation Reduction Act was signed, it has a huge amount of money for new battery manufacturing in the United States, but there's not a lot of workforce development in US institutions. So this is where this new battery lab comes in. And there we teach two classes, ENMA 416 and 417, which I helped develop within the department. And we basically use the lab. Along the right side there is basically the entire battery manufacturing process. So within the lab, students, undergraduate students, can go through each of these steps and get hands-on experience, which they can then utilize in an actual manufacturer. So this is a cell making schedule that we teach to the students. So in the lab, we basically do this, or in the class, we basically do this about 4 times. And since the class is within the material science department, we focus on studying different cathode chemistries and anode chemistries. So we're making new electrodes every time. So it starts off with the slurry mixing step, which is maybe the most important step to make the good battery. So in this step, you'll have, in the cathode or the anode, you'll have an active material. So the students have done lithium iron phosphate batteries, nickel manganese cobalt batteries, and lithium manganese oxide, and studied how those voltage characteristics change with different chemistries, studied how the aging properties change. And these are the same characteristics that's going to be important for working for SAFT, which is one of the Maryland manufacturers we're working on in a bigger kind of gigafactory scale, is understanding how those chemistries play out. So we mix and cast the slurries on our electrodes, and we have to dry those overnight. For this class, we basically have 2 totally separate lines. One is for anode manufacturing and one for cathode. So on our anode side, we're all water-based. So that's the industry standard nowadays to get away from NMP, which is very toxic, but we do use NMP in methylpyrrolidone for the cathodes. Day 2, we can calendar and punch the electrodes out. So one of the coolest things about this lab is that we're making pouch cell batteries. So when you look in your cell phone or your consumer electronics, you're more likely to find a pouch cell rather than a coin cell, which is what's normally kind of used in academic research. So we're stacking those electrodes. We have a very nice cell stacking machine. These are about 5 by 5 centimeter cells. And then we take them into the glove box for the final sealing and the cell testing the next day. So we also do field trips to the manufacturers. So here's the class at SAFT. We actually can go in their dry room and see what they're doing. And I've only taught the class twice, and one of my students now works at SAFT, so we get to interact with him and form that relationship between UMD and SAFT. And hopefully continue to bring that up. So just want to acknowledge the MSc department And then these classes were developed with some people from the US Navy, Daphne Fonavia and Julie Simmons, who've been really helpful, giving their time and effort for that.
Thank you, Dr. Cisco. Now we have Dr. George Alexander, who is a visiting assistant research scientist in the Department of Material Science and Engineering here at UMB. His research focuses on solid-state batteries, and he's passionate about making meaningful contributions to the scientific community while inspiring future generation of researchers. Welcome.
Thank you for having me here. And so there's a great talk all about having agriculture, harvesting. I'm not into that field, but then I really enjoyed that talk. But then a little bit into the sector of having energy's importance there. The previous speaker has already talked about what's the importance of having, kind of an energy storage device here, right? So I'll be talking a little bit about why energy and what kind of energy storage devices that we can use. So moving forward, a little bit of animation about like energy plays an important role in every aspect of human life. And we mainly depend upon fossil fuels like coal, oil, and natural gas. And towards the right, you could see a graph where it shows at 2025 where we are, and the natural gases are depleting at an alarming range. And in 2050, it's going to be at even lower rate. So one of the most important way of having this energy storage, how can we do it, is through renewable. So we can have energy through sun, we can have energy through water, we can have energy through wind. So, but then the main problem of all these renewable energies are, they are intermittent, because we need in one way to store this energy. That's where the importance of an electrochemical energy storage device comes. So that is where we are working on to have safer and cheap batteries. So the other important thing is the oil consumption that we have, because if you see, majority of the oil consumption comes from the road transportation. We have industrial and still agricultural still uses the oil consumption part of it. So this is a graph where it shows when you have more oil consumption, there will be greenhouse gases being produced like carbon monoxide, carbon dioxide. You can see a graph which the brown color shows where it should be, and the blue color shows where we are now. So 2025, we should be emitting only like 2,000 million tons of carbon dioxide, but then we are already emitting 3 times more of it. So the use of carbon dioxide or carbon monoxide is like creating a lot of climatic changes, and it's important to have an energy storage device from there. And I have put an electrical car here because since most of it's coming in from the road transportation, that's why I put an electric car, but then energy storage device could help us in agriculture sector, industrial sector, road transportation. A little bit talk about different energy storage device, since we have lithium metal, which has been a lot of importance. Why lithium metal or lithium-ion batteries? Because of the energy density that we have. Because if you could see, energy density is nothing but how much energy a system can store per se of certain mass. So lithium-ion batteries are very high energy densities, and in a battery, you see you have a cathode and an anode, and when you charge the cell, what happens? Lithium ions move from the cathode to the anode, and then when you discharge it, it moves like on the opposite direction. But the main problem that we have in the present lithium-ion batteries is safety. Right, the batteries catch fire, and that's the main thing that our focus is on. So here it's an image that shows a kind of a tree-like growth that happens through the anode, passes through the liquid electrolyte and touches the cathode. We know that when a positive and a negative joins together, boom, catches fire. So that is where the importance of a solid state system comes, where you have a solid system that replaces a liquid system. So now you don't have that enough space for this kind of tree-like growth that comes from the anode to go towards the cathode, and makes it more safer batteries. So we can have safer batteries, but the other importance is whether we can have cheap batteries. We depend upon a lot of nickel, cobalt, manganese, and all. So is there any other chemistries in the world that we can use which is cheaper? One is sulfur, because if you could see the sulfur's abundance is much higher than that of nickel and cobalt. The other things are like sodium and air. These are off the chart. I didn't have to add it here, but then they give very high capacity. Capacity in the sense, what I mean is energy density. So if I have higher energy density, which means I can store more energy. So this is, people have seen with using air as a cathode and sulfur as a cathode, they could store more energy. But then the main issue with these systems was when they had a liquid in there, the oxygen or the air, the sulfur reacts with the liquid and it kinds of impedes the reactions. Because if you want a battery, you need to run it for years, right? You don't want a battery that runs for a couple of cycles and then die off. So that is where another important comes, that when we have a solid system in there, which doesn't react with any of these, like air, sulfur, sodium or something, it kinds of increase the stability of your cell. So, to have an ideal solid system, you should have, these are the characteristics in a electrochemical perspective, we say about conduction, but then it's all about, you should have smooth lithium ion motion happening in a system. And also it should be like really comparable to what the present liquid electrolyte system has. So we have in our lab, we developed new solid electrolyte systems. These are called garnet-based solid electrolyte systems. This is a state-of-the-art solid electrolyte. So we developed that in our lab and then kind of studied this. But when you say about solid state batteries, though it improves the energy densities or the safety of the system, the major problem comes from the interface. Someone said previously that bulk was invented by God and the surface was invented by evil, because all the problems in material chemistry comes from the surface. So that is where when you imagine, when you have three systems, which is a solid electrolyte and anode, these are like three bricks kept together. So when you have three bricks kept together, the motion between the cathode and the solid electrolyte and the anode will be impeded. So a lot of focus, which is like really trending now in 2020s in our lab, we have developed though we have developed new solid electrolyte system, we also tried to resolve these issues, what happens at the interface between the cathode and the anode. So these are a kind of a nutshell about what we do in our lab and what kind of research we do, and could help in motivating why we need more energy storage devices. With this, I would like to conclude my talk, and thank you.
Thank you, Dr. Alexander. Now we have our last speaker, Taryn Sudol. She is a Coastal Resilience Coordinator at the Maryland Sea Grant. She received her Master's of Science in Conservation Biology and Sustainable Development from the University of Maryland. Since 2018, her work has focused on promoting a greater understanding of the ecological and social effects of sea level rise. She works with scientists, land managers, government agencies, community members, and other stakeholders to plan for adaptation measures and actions. So I would like to welcome Taryn Sudol.
Hi, thank you. I appreciate everyone sticking around for my talk. We'll round things out with sea level rise for all the water issues that we heard today. For those of you unaware, Sea Grant is like the sister of Land Grant Extension. We focus on marine and coastal issues, as you just heard. And so Sea Grant is not agriculture, but we are sea level rise. And we know that on our coastal lands, some of which are in agriculture, we're seeing tidal regime shifts. We're seeing greater nuisance flooding events, some of which extend pretty far inland on a sunny day, or are, very much affected by storm surge when a big storm comes around. And not only that, there's the saltwater intrusion issue from the groundwater. So in essence, farmers are experiencing wetter and saltier lands when they're located closer to the shore. And so Maryland Sea Grant, in a study that I was able to conduct, we wanted to know what are agriculture list producers, what are they experiencing, and what is their decision-making process given these environmental forces that are happening. And so what we did was a series of interviews, virtual workshops that delivered resources and lectures from researchers, as well as connected farmers to themselves, to one another, and then associated pre and post survey results. And so what might come as no surprise is basically, as I just mentioned, soils are saltier and wetter. You'll also note that of the participants who completed the survey, all of them are experiencing some change. And I think the results in the middle here, you can get a closer look at them on the poster, are these associated shifts in vegetation, and with changing vegetation, changing wildlife. And so they're experiencing a lot new destructive wildlife that can be a big impact to their crop productivity, a lot of crop damage, as well as just labor and control, and even encountering policies that don't help them manage either the nuisance vegetation or wildlife that are affecting their properties. I'll also point out the ditch maintenance is a huge factor that a lot of farmers contend with. The ditch is originally meant to drain lands and now serving as a conduit for these flooding events. And so just to boil this down really fast, and I'm really glad to dwell on this further in our next session, we asked them, what are you trying to do in the short and long term? And so when we're thinking about sea level rise in communities, a common framework, particularly on national public natural lands, is, are you able to resist this environmental forcing, in this case, sea level rise? In the farm on the coast, this may mean tide gates, it may mean berms, it may be living shorelines in order to stop that water from reaching their lands. I think this is a function of exposure. If you are a small enough farm, you might be able to afford these interventions, but if you have a pretty significant coastline, putting in any of the, at least the berms or the living shorelines, can be pretty cost prohibitive. Secondly, you can accept that water is going to be on your landscape and work with that water. That might mean changing your crops to more flood and salt tolerance, or it might mean patches of your land go fallow. And why a farmer may want to do that is a pretty interesting question. Maybe they just don't know what to do with that patch that is no longer a profitable investment for crops, or they feel like someday they'll be able to do something with it, or they really don't like the idea of the other options available, perhaps through government assistance programs, which leads to the final category, which we call direct, which is the idea that you're no longer going to farm this affected piece of land. Instead, you're going to treat it as a new land use. And primarily, our go-to suggestion is wetlands. And so that might be appealing in terms of the conservation easements available, or to use as a recreation source, such as hunting. But there is a lot of resistance perhaps to government programs, the idea of loss of control being very unappealing to some, as well as some frustration on the inflexibility of some of the currently designed programs. So with all that in mind, here are the results. And to sum it up, basically in the short term, folks are going to continue to resist or accept this sea level rise affecting their property. But in the long term, they say they're going to direct, they're going to change their land usage. And so in some ways, this opens up opportunity, but the someday is something I'd like to narrow in on further. What are these threshold events that lead to transitions? And so really curious and hopeful that this conversation and research can continue so that we can offer support to these farmers and producers as they're facing this really difficult adjustment and change to their long-term or aspirational or generational career. Thank you.